Greetings everyone, and today we are going to discuss about a new chapter prokaryotes. Before discussing prokaryotes, we need to first discuss what is taxonomy of prokaryotes. Basically, prokaryotes, as we previously known, that what are prokaryotes? These are those organisms which are, you know, having which are not having cellular organelles, as well as these are primitive organisms known before karyote means before nucleus, which means they do not have nuclear membrane. They have a primitive without nuclear membrane. Even so, they do not have nucleus because they only have genetic material which is found around a specific part of cytoplasm, which is known as a nucleoid region. Hence, they do not have nucleus, so these are before nucleus. Apart from that, eukaryotes, we are previously known about eu means true and karyon means nucleus. They have a true nucleus. Apart from that, when it comes to the taxonomy of prokaryotes, let's discuss that here. So previously, initially in the early centuries, the animals were classified into two major classes and into two major kingdoms. Actually, it was before the kingdom classification, so it was only categorized into two groups. We can also call this as a class or group or kingdom. But here, I'm just generally talking about two groups. This was done by Aristotle, and here he classified organisms into two groups, which first involved animals and second involved plants. Now, when it comes to animals, he classified animals with blooded animals and without blood animals. On the other hand side, when he classified plants into herbs, shrubs, and trees. So this was a basic classification at that time, but this was not sufficient for a wide and diverse range of classification of different organisms, because organisms present within the biosphere are diverse everywhere. Within the biosphere, we are within on the earth, there are organisms which are not only plants or animals but other kinds of species as well, such as we are having fungi, we are having algae, we are having protists, we are having different kinds of prokaryotes. So at that time, uh, this was not sufficient, as a result of which, uh, a scientist who came, who is also known as father of taxonomy, Mr. Carl von Linus. It has its original name, but he actually cut his name down and marked it as Carolus Linus, because he introduced binomial nomenclature, as a result of which he marked his name as Carolus Linus. And w- as he marked the classification system of binomial nomenclature, he then also utilized uh, a first step. He took the first initiative towards creating a classification system, which was a two kingdom classification system. It's given over here as well. Before discussing that, we need to understand what does the word taxonomy means. Taxo means group. Taxo means group, whereas nom means name. Grouping on the basis of grouping on the basis of categories. No means names, right? Naming on the basis of groups. No, or in other words, we can say that naming on in form of groups is known as taxonomy. Apart from that, uh, as you can see within this, we are going to first discuss our main part, which is the two kingdom system, in which the organisms, according to Carolus Linus, were classified into a kingdom Animalia and kingdom Plantae. And organisms in kingdom Animalia involved those which were without cell wall, and plant kingdom which were with cell wall. So uh, all the animals with cell wall were classified into animal kingdom, which involved the example of all the animals which are present nowadays in kingdom Animalia, as well as protists which were without cell wall. Apart from that, plant kingdom involves all the organisms with cell wall, including fungi, algae, and other types of protists as well. So that also had some subkingdoms: Cryptogamia, Phancrogamia, Halophyta, Bryophyta, Trichidophyta, and Gymnosperm, Angiosperm. These were some classification over which it was performed. And at that moment, bacteria was classified into the phylum Thallophyta within the plant kingdom, and within the subphylum Schizophyta. So this was the origination basis. Apart from that, uh, when it comes to some other parts,
which I would like to discuss over here is about the third kingdom classification, because a Carolus Linus classification, which was purpose in 1758, was not adequate to classify organisms, because all organisms had some sort of dissimilarities within their respective kingdoms. As a result of which, there was a three kingdom classification system which was purposed by Ernest Haeckel and John Hogue. In some books, it's written as John Hogue as well. So Ernest Haeckel basically in 1866 classified the unicellular organisms from both Animalia and Plantae into a separate kingdom which was known as Protista. In a separate kingdom which was known as Protista, all of the unicellular organisms were placed here. After which, uh, when there was a discovery of Edward Chatter. Edward Chatter basically divided all of the organism into two major empires, prokaryote and eukaryotes, such that prokaryotes had the characteristic of we have previously discussed that prokaryotes will do not have a proper nucleus, or they do not have a nucleus at all, as well as they do not have cellular organelles. They lack cellular organs, but eukaryotes are having two nucleus as well as cellular organelles are present over there. So prokaryotes and eukaryotes were when prokaryotes and eukaryotes were uh, classified. So on the basis of that, uh, it was able to classify organisms. One more thing, that here, uh, um, when it comes to when it comes to a two kingdom classification system, it was on the basis of morphology and morphology and some aspects of cytology as well. But the areas which were lacking involved body organization, whether it is multicellular or unicellular, as well as it didn't have mode of nutrition, as well as mode of nutrition, mode of nutrition, body organization, as well as whether they are autotrophs or heterotrophs, and if heterotrophs, ingestive or absorptive heterotrophs. Moreover, it also involved genetic barrier, as well as the genetic cytology, genetics, physiology. All of these were not considered, as well as uh, in accordance with those, molecular biology was also considered. So these were the areas which were lacking in Carolus Linus two kingdom classification system. Apart from that, when it comes to the three kingdom classification system, it had classified organisms on the basis of body organization, but other aspects were also remaining. Now, when eukaryotic and prokaryotic organisms were introduced, Edward Jenner introduced the two empire system, which was eukaryotic and prokaryotic. Now, at that moment, Copland in 1956, basically, he classified prokaryotes, which were previously placed in kingdom Protista, because Protista at that moment included unicellular prokaryotes as well as unicellular eukaryotes. So at that moment, these unicellular prokaryotes were separated out by Copland, and allowed Copland to create a separate kingdom for prokaryotic organisms, which was known as Monera. So prokaryotes were first given their uh, separate kingdom by Copland in his four kingdom classification system, and as a kingdom Monera. After that, when there was Mr. Robert H. Whittaker. Whittaker came, and Robert H. Whittaker basically classified the organisms in 1969 on the basis of the given factors which are present within these areas. He considered body organization, whether they are unicellular or multicellular, unicellular prokaryotes, unicellular eukaryotes, multicellular eukaryotes, mode of nutrition, autotrophs, ingestive heterotrophs, or absorptive heterotrophs. cytology, that what is the cytological constitution, molecular biology, and these factors were considered. So this is how it was all considered, and then he classified organisms into four major groups. Previously, kingdom fungi or kingdom mycota organisms were placed in kingdom plantae. But after that, when it came to Robert H. Whittaker, he saw that genetically, Robert H. Fungi has similarities with both plant and animal. So, what are these similarities? Let's discuss that as well. It is having cell wall, which is the similarity with plant, and it does not perform photosynthesis. Does not perform photosynthesis. Apart from that, uh, it is having centrioles, which is a characteristic of characteristic of animal. Sorry, centrioles and no act of photosynthesis was a characteristic of animals, while cell wall is a characteristic of plants. Moreover, another characteristic of plant present in them was that these were having cell wall. These were having cell wall as well as cell wall was not cell wall was present there. That was a characteristic of plants, as well as they had a characteristic that they were dependent. They had the characteristic that they were non-motile. They were non-motile. Unlike animals, these were non-motile. So as they were non-motile, they were also considered in this kingdom. Now, when Carolus Linus found a genetic constitution of fungi, it found that genetic constitution of fungi was linked with both, does not link with both of the kingdoms, which means it mostly linked with animals, but as it was, quite unique, it then, instead of putting it in any of those kingdoms, he put it into a separate kingdom, which was known as Kingdom Mycota. Now, at that moment, his main criteria was cellular organization, mode of nutrition, and reproduction. At that moment, this is how he classified it. And at that moment, these are the five kingdoms which are present over here. Some characteristics are also written, just a bit blurred. Moreover, apart from that, when his classification was done in 1988, there were two scientists named as Line Margulis and Carlin Schwartz. Line Margulis and Carlin Schwartz, who basically modified the Whittaker's classification system. According to Whittaker's classification system, algae is an organism, multicellular algae. Multicellular algae were placed in kingdom Plantae. Multicellular algae are placed in kingdom Plantae, but uh, according to Margulis and Schwartz, multicellular algae has a dissimilarity with plants, such that plants form embryo after the formation of zygote, but algae was an organism which formed body directly after the zygote, which means it didn't have the stage of embryo, it directly formed body from the zygote. As a result of which, as this stage was missing, it showed that this is a dissimilarity between algae and plants, due to which they decided to put multicellular algae into protista, and at that moment they renamed this terminology protista into protoctista. At that moment they renamed it as Protoctista, this kingdom as Pro Protoctista, which means ragbag material, such that all of the ragbag material of all the eukaryotic kingdoms were placed in kingdom Protoctista. So at that moment, this was named as Protoctista due to its ragbag material. Apart from that, Algae. Algae basically is, on the other hand side, if we consider Whitaker's classification is in a way correct because it gives us the idea that multicellular algae and unicellular protists cannot, have a similarity because they are, on the basis of their body organization, both are different. So in this case, this was, a bit of the classification, but we are going to follow the Margulis and Schwartz classification, such that algae is placed in kingdom Protoctista, which is named as Protoctista. Apart from that, after that, Carl Voici and his co-workers had introduced a new system, a new taxon, which was known as domain. Domain system was one which was higher at a higher rank than kingdom system, and at that moment, as it was at a higher rank than kingdom system, it basically classified all of the organisms on the basis of their subcellular components, such as their, you know, uh, classified organisms on the basis of their subcellular components, including ribosomes as well as lipid bilayer. So how it classified in ribosomes? We know that within the rRNA present within the ribosomes, which has a conserved gene. That conserved gene has no mutations, which means it can, remain within its same genetic constitution without any mutations for a longer period of time. As a result of which, when these were, when they were fossilized, when these genes were, retraced with the genes of other organisms, it was found that bacteria and archaea were, in a different kingdom, different domain. So how? um, Basically, we are having two major domains of bacteria, which includes archaebacteria and eubacteria. Here archaebacteria was the one which involved archaebacteria, which involved basically the conserved gene present within the rRNA of the ribosome was having a different sequence of nucleotide than the conserved gene of rRNA of the eubacteria. As a result of which, both of the genes being different, different sequence of nucleotides, it showed that a class, I mean, we cannot call it as that, but a certain group of bacteria is having dissimilarity with bacteria, and that is known as archaeobacteria. And one more thing, when it comes to lipid bilayer, archaeobacteria are having their lipid bilayer structure, phospholipid bilayer structure, with ether linkage, whereas bacteria are having eubacteria or bacteria are having their linkage with l- ester linkage. Now, this was also a consequence. So, as archaebacteria were having the ester linkage and eubacteria were having the ether linkage, archaebacteria were having a weaker, ester linkage, because ester linkage is a weaker bond, and bacteria are having ether linkage, which is a stronger, bonding. So that ether linkage was stronger in bacteria. Apart from that, when it comes to uh, their classification, it was on the basis of the three evolutionary lineages of organisms that evolved on Earth, such that, according to Carl Voici, the first organisms to evolve on Earth were the archaebacteria, after which eubacteria and then eukarya were there. So, uh, this classification divided the kingdom Monera into two major domains, which includes archaeobacteria and bacteria. Archaeobacteria are basically having the following classes, following phylums as well. You can just look into it. We are going to discuss that hopefully in our next video. For now, this is eukarya. Eukarya is divided into four of its kingdoms, such as Protoctista, Animalia, Plantae, and Mycota, while Monera involves all of the bacteria, while archaeobacteria had a primitive uh, domain of bacteria. So this is basically a brief intro about all this, and hopefully we'll meet again in another video.